हॅलो स्टुडंट्स, रिप्रोडक्शन इन प्लांट्स चॅप्टर मध्ये आपण स्टडी केले मेन इव्हेंट फर्टिलायझेशन. आफ्टर फर्टिलायझेशन zygote converted into embryo and primary endosperm nucleus converted into endosperm. डेव्हलपमेंट ऑफ इंडोस्पर्म हा पार्ट आपण स्टडी केलेला आहे आणि आज आपण स्टडी करणार आहोत डेव्हलपमेंट ऑफ इम्ब्रियो. डेव्हलपमेंट ऑफ इम्ब्रियो also known as embryogenesis. Here in word embryogenesis, genesis means formation, तयार होणे. एम्ब्रियो तयार होणे त्याच प्रोसेसला काय म्हणतात? एम्ब्रियोजेनेसिस. What is embryogenesis? The process of development of zygote into an embryo is called embryogenesis. Embryogenesis takes place step by step from zygote. First step आपण स्टडी करणार आहोत. Zygote divides to form two cell proembryo. After fertilization, zygote is formed and zygote divide cell, and that two cell structure known as a proembryo. Proembryo means primitive embryo. First structure just ते zygote विभागून दोन सेल तयार होतात त्या स्ट्रक्चरला प्रोइम्ब्रियो म्हणतात. सुरुवातीचा एम्ब्रियो. Zygote divides unequally. One cell is larger and another cell is smaller. Larger basal cell is present towards the micropyle end is called suspensor initial cell, and another smaller cell towards the chalaza is called terminal cell or embryonal initial cell. Second division, suspensor cell divides transversely one plane, produce filamentous suspensors of 6 to 10 cells. Means these suspensor cells divide transversely by mitosis, and after division and redivision form the suspensor. In suspensor, the first cell is toward the micropylar end, becomes swollen and function as a posterium. See diagram. Larger swollen cell known as a posterium. First cell of suspensor toward the micropylar end or end cell of a suspensor swollen and is known as a posterium. सस्पेन्सरच्या टोकाची कंपॅरेटिव्हली लार्ज अँड सोलन फुगलेली आढळते आणि त्या सेलला टर्म यूज केली जाते पोस्टेरियम. अँड द लोअर मोस्ट सेल ऑफ सस्पेन्सर इज नोन ॲज अ हायपोफायसिस. सस्पेन्सरच्या एंडच्या सेलला होस्टेरियम म्हणतात आणि जो इम्ब्रियोला अटॅच असलेली सस्पेन्सरची एंड शेवटची सेल आहे लोअर मोस्ट सेल ला त्या ठिकाणी हायपोफायसिस. सस्पेन्सर हेल्प्स इन द इम्ब्रियो इन दी एंडोस्पर्म. सस्पेन्सरचे फंक्शन आहे सस्पेन्सर पुश द इम्ब्रियो इन दी एंडोस्पर्म. एंडोस्पर्म मध्ये इम्ब्रियोला पुश करतो जेणेकरून हा वाढणारा इम्ब्रियो त्या ठिकाणी अन्न एंडोस्पर्म मधून जास्तीत जास्त. थर्ड डिव्हिजन आहे द इम्ब्रियोनल इनिशियल अंडरगोज 3 सक्सेसिव्ह मायटोटिक डिव्हिजन टू फॉर्म दी ऑक्ट. Suspensor initials form the suspensor part, then embryonal initials form the embryo. Primitively, this embryonal initial divides and redivides form octant. After first division, one cell divided into two. After second division, 2 cell divided into 4. After third division, 4 cell divided into 8 cell. And 8 cell stage is known as a octet. Here, all these divisions are right angle to each other. In embryonal development, lower tier of 4 cells of octant gives rise to hypocotyl and radicle, whereas 4 cells of upper tier forms the plumule and the 1 or 2 cotyledons. Here, lower tier of 4 cells means lower region of octant rise to hypocotyl and radicle, and upper tier forms plumule and 1 or 2 cotyledon means octant 8 cell embryo मधील upper region मधील 4 cells 1 or 2 cotyledon structure तयार करतात. Monocot 1 cotyledon structure form, dicot 2 cotyledons form from the upper tier of octant. The hypophysis region further divide and gives rise to part of a radicle and root caps. Hypophysis region of suspensor divide and redivide forms many cells, and that cells take part in formation of a radicle and root cap. After formation of a octant embryo, further these 8 cell divide and redivide forms the heart-shaped embryo. The octant embryo, it is globular embryo, and then the upper tier of octant divide in several planes and becomes heart-shaped, and that embryo known as a heart-shaped embryo. This heart-shaped embryo further forms the 2 lateral cotyledons and terminal plumule. Further enlargement of a hypocotyl and cotyledons results into the curvature of embryo, and it appears as a horseshoe shape. Heart-shaped embryo further enlarge and forms curvature embryo, and it appears horseshoe shape. Embryo development in dicot and monocot is same or similar up to the octant stage. And dicot मध्ये embryo development ही octant stage पर्यंत सिमिलर सारखी असते. नंतर मात्र after octant stage dicot 2 cotyledons are formed, and in monocot plant mono single cotyledon develop. Structure of a monocot seed. In monocot embryo, single cotyledon occupies terminal position and the plumule is lateral. Here, the single shield-shaped cotyledon is called as a scutellum. Surrounding to the cotyledon, the presence of a covering, and it forms a single shield-shaped cotyledon, and that structure called as a scutellum. The protective sheath of a plumule is called as a coleoptile. Surrounding to monocot embryo, presence of a protective sheath, and that protective known as a coleoptile. Coleoptile, it is a specific characteristic of monocot embryo. It's present only in monocot seed or embryo and absent in a dicot. In monocot embryo, also the presence of a radicle, and radicle gives rise to the root, and that radical part also known as the coleorhiza. Plumule covering known as a coleoptile, and the covering protective sheath of a radicle known as a coleorhiza. Rhiza root, so that here term use coleorhiza. So that after fertilization, zygote converted into embryo and primary endosperm nucleus converted into endosperm. Complete जर structure study केले तर in plants finally ovule is converted into seed and ovary converted into fruit. Embryogenesis in plants हा टॉपिक आपण ॲनिमेशनच्या मदतीने पुन्हा एकदा रिवाइज करूया. व्हॉट इज embryogenesis? Formation of a embryo or development of a embryo known as a embryogenesis. Embryogenesis, step by step mitotic division takes place. Here, after fertilization, zygote divided into two cells, unequal cells, that is asymmetrical cells. Smaller cell, apical cell, also known as a terminal cell, embryonal cell, and larger basal cell, microbial end cell, known as a suspensor cell. Here, apical cell, embryonal initial cell, gives rise to embryo, and lower cell gives rise to suspensor. Lower basal cell, suspensor initial cell, divide and redivide transversely and form 10 cells structure, that is suspensor. Suspensor push the embryo inside the endosperm for nourishment of an embryo. Lowermost cell of a suspensor towards the embryo, known as a hypophysis, and end cell, swollen and larger cell, known as a posterium. Basal cells gives rise to suspensor, and apical cell, that is embryonal initial cell, gives rise to first octant stage embryo cell front and 4 cell back side. Then after globular stage, heart shape embryo is formed. Octant stage proembryo divide and redivide mitotically, form heart shape embryo. Heart shape embryo further gives rise to different parts of a plant. Here, apical region gives rise to the cotyledons and shoot apical meristem. The middle region gives rise to the hypocotyl, and the hypophysis region gives rise to the radicle and root system. Further, next stage of a embryo is horseshoe-shaped embryo. Last stage, horseshoe-shaped embryo is horseshoe shape due to the curvature of a cotyledons and mature stage of a embryo.